My lab at the Eunice Kennedy Shriver National Institute of Child Health and Human Development is not on the main campus in Bethesda, but rather out in the Maryland countryside near a little town called Poolesville. So when I talk about the Poolesville colony, that's our colony of approximately 400 rhesus monkeys that we maintain, many of them in outdoor, free-ranging outdoor enclosures. And since its inception, my lab is focused on the study of individual differences in personality or temperament in these monkeys and the biological substrates that underlie these individual differences, and in particular how genetic and environmental factors act, and we think much more importantly actually interact to shape individual developmental trajectories. Over the years, we've been particularly interested in two subgroups of monkeys within this population, as well as at two field sites that we've had the opportunity to utilize over the years. One subgroup comprising about 20% of the monkeys at Poolesville and about 20% of the monkeys at these two field sites seem to be unusually fearful or anxious in the face of novel or mildly challenging circumstances. So situations and stimuli that most monkeys find really interesting and will readily explore, these monkeys try to avoid, and if they're forced to confront these situations, they show profound arousal of a variety of biological systems. The system that we've spent the most time looking at is the hypothalamic pituitary adrenal axis, and we see in these monkeys prolonged elevations of cortisol, whether we're measuring it in blood or saliva or more recently in hair. These monkeys look very much like a subgroup of human children that people like Jerome Kagan and Nathan Fox have been studying over the years, and Tom as well. These are children who Kagan calls behaviorally inhibited. They show many of the same behavioral and biological features as our uptight monkeys, and we now know that these children are at risk for developing childhood anxiety and depressive disorders, and for some of whom this risk carries over into adolescence and adulthood. There's a second subgroup of monkeys, maybe 5 to 10% of the population at Poolesville and 5 to 10% of the population in the wild, who seem to be unusually impulsive, and their impulsivity gets them into trouble. They do stupid things that other monkeys know better than to try, like get between a high-ranking female and her offspring, or they may repeatedly attack a dominant adult male, and when that male beats them back, they're right back in that male's face. So not surprisingly, they tend to generate a lot of aggression from others in their group, and they themselves are unusually aggressive as well. They also have biological characteristics that seem to differentiate them from others in the group. The characteristic we spent the most time looking at is an apparent deficit in serotonin metabolism that we see in terms of chronically low levels of the primary central serotonin metabolite 5-hydroxyinylacetic acid, or 5-HIAA. And these monkeys also, this subgroup also resembles a subgroup of children that people like Richard Tremblay have been studying over the years. These are children who already by 2 years of age have been identified as being highly aggressive. The high levels of aggression are maintained throughout development, so by the time they hit the school system, they're often causing classroom disruptions. They're often diagnosed as having externalizing disorders, and at least in the U.S., you can find many of them by the time they're in their mid or late teens, either in prison or in the morgue. Now, whether a monkey turns out to be aggressive or impulsive or something else, virtually they all start the same way, at least in the wild. All rhesus monkey infants spend virtually all of their first month of life in intimate physical contact, or at no more than arm's length away from their mother, their biological mother, and during this time, a strong and enduring attachment bond is established between mother and infant. It's a bond that's functionally equivalent to the type of attachments that human infants routinely form with their caregivers in virtually every culture in the world. And then, once this attachment has been formed, beginning in the second month of life, these monkey youngsters start to explore their environment in a very stereotypic fashion. They leave their mothers for short periods of time, then come running back to their mothers for a period of contact, go out and explore a little bit more, run back to their mothers for a period of contact. It's almost as if they're attached to their mothers by a long rubber band, and as time goes by, the time spent away from the mothers becomes greater, so that by 6 months of age, they're spending only about 20% of their waking hours in actual physical contact with their mother, as opposed to the 100% they were spending in that first month. Yet the mothers remain a very important part of their social world, because as Bowlby has taught us, if you lose access to your secure base, any motivation to explore will rapidly disappear. As these monkeys spend more time away from their mothers, they begin to interact with other members of the group, and increasingly these interactions involve peers, other monkeys the same age as the monkeys, youngsters themselves. And in the wild, this is not by accident. In the wild, these monkeys are seasonal breeders, so that although they copulate year-round, 80 or 90% of a troop's infants are born within a 2 to 3 month window. And what this means is any youngster born during this time will have plenty of other youngsters who are about the same age and cognitive and physical and social skills as themselves, and that's who they choose to hang out with much of the time. So through the rest of the first year, the childhood years, the rest of the first year of life, the second year of life, and the third year of life, these monkeys will spend several hours every day in active social play with their peers, and it's in the context of this play that virtually every behavior pattern that's going to be important for normal adult functioning can be developed and practiced and perfected long before it actually has to become functional. And this is especially true for reproductive behavior, and it's also especially true for the socialization of aggression that comes into these monkeys' repertory at between 4 and 6 months of age. And work by my mentor, Harry Harlow, many years ago, as well as more recent work in the Netherlands, has demonstrated quite convincingly that rhesus monkey youngsters who are otherwise well socialized but denied the opportunity to play with peers when they are growing up inevitably grow up to have real problems with reproduction and real problems controlling aggression. Now, not all monkeys at Poolesville are raised this way. Some of them are cross-fostered to unrelated females at birth, so we can separate out genetic and environmental factors, and others are reared without parents or without mothers using a procedure designed by Harlow many years ago called together-together or peer-only rearing. Here, infants are separated from their biological mother at birth well before any kind of attachment can be established. They're hand-reared in a neonatal nursery for the first few weeks of life, and they're then put into small peer groups containing 4 or 5 monkeys like themselves, where they stay with one another 24 hours a day, 7 days a week, until they're 6 or 7 months of age. At this point, the peer-reared monkeys are moved into larger social groups that also contain their mother-reared counterparts, who have been weaned from their mother at this age, much as happens at that point in the wild. And thereafter, mother-reared and peer-reared monkeys are all living in the same physical and social environments. So all of the rearing condition differences that I will be referring to can be traced back to that initial 6 or 7 months of life. And when you rear monkeys like this, they typically develop hyper attachments to one another, intense attachments, but these attachments, while very strong, are actually dysfunctional because these youngsters spend so much time clinging with one another instead of going out and beginning to explore their environment like their mother-reared counterparts would be doing. And when it gets time to the age where they should be starting to play, they do indeed play, but the play never reaches the level of sophistication or complexity that you see in their mother-reared counterparts. And when you think about it, it's not surprising because these monkeys have to serve as playmates and attachment objects at the same time, and they don't do a great job of either. So even though these monkeys are with potential playmates 24 hours a day, they are actually play deprived as they are growing up. And this rearing has some long-term consequences. For one thing, monkeys reared this way grow up to be more fearful than you would expect had they been reared by their biological mother. And behaviorally, they look very much like that 20% of monkeys in the wild who seem to be unusually fearful or anxious in novel circumstances. And like that 20% of monkeys in the wild, they show higher levels of cortisol under comparable challenge conditions than their mother-reared counterparts. Something else happens to these youngsters as they're growing up. They become not only more fearful, but more aggressive. And they start developing these patterns of impulsive aggressiveness that you see in that 5 to 10% of the population in the wild. And like that 5 to 10% of the population in the wild, they show deficits in serotonin metabolism, whether we're looking at the first year of life, when these differences are significant, or even later in life, this should be year 5, when the levels have dropped for everybody, but if anything, the rearing condition difference has gotten larger than it was before. We have done studies in collaboration with my colleagues in the Alcohol Institute that show that under a happy hour situation, peer-reared monkeys consume as a group more alcohol than their mother-reared counterparts. We've done more recently, we've done neuroimaging studies, both PET scans and structural MRIs. Here are findings from a PET scan study we'll just focus on the left, looking at serotonin binding potential. What you have here is a composite mother-reared, front view of a mother-reared versus peer-reared. top view of a mother-reared versus peer-reared composite, side view of a mother-reared versus peer-reared composite, and simple inspection suggests that the peer-reared monkeys' brains are not lighting up as much as their mother-reared counterparts. And when you look at the actual data, indeed that is the case throughout the brain, whether you're looking at the raphe, the thalamus, the striatum, the temporal, or the frontal regions. Peer-reared monkeys have significantly less serotonin binding potential than their mother-reared counterparts. We recently published a paper looking at serotonin 1A receptors. We see the same pattern of rearing condition differences. We also recently published a structural MRI study, and once again, peer-reared monkeys' brains are structurally different in significant ways than their mother-reared counterparts. Very recently, and not yet published, are some data looking at genome-wide methylation patterns. These are studies done in collaboration with Moshe Ziff and his group at McGill, and my ex-postdoc, Allison Bennett at Wake Forest, now at Wisconsin. And what we have here are Moshe's genome-wide methylation patterns, chromosome by chromosome, looking at, on the top line, prefrontal cortex, and the second line, lymphocytes. These samples were obtained when the monkeys were full adults, 8 years of age, differentially reared only for that first 6 months of life, and thereafter under the same conditions. And each of these bars is an individual gene. If it's above the line, it's more methylated in mother-reared. If it's below the line, it's significantly more methylated in peer-reared, both in prefrontal cortex and in blood. Chromosome 2, the same thing. I could go through all the chromosomes. The bottom line is that there are an enormous number of genes that are differentially methylated as a function of those first 6 months of differential early social experience. In all, over 4,400 genes, both in blood and in brain, are differentially methylated as a function of early experience, 1/5 of the entire rhesus monkey genome. And as Michael Kober pointed out, there is certainly some tissue specificity, but there are also some areas, some genes, where the patterns are exactly the same in prefrontal cortex as they are in lymphocytes. Here and here, 30% of those 4,400 genes have this similar pattern. Another 25% of those 4,400 genes have the opposite pattern. That is, if it's more methylated in mother-reared in brain, it's more methylated in peer-reared in the lymphocytes. So these early experiences are having clearly epigenetic effects. And this is methylation patterns, but as you all know, methylation is not the same as expression. Here are gene expression data from Steve Cole at UCLA. Here's a heat plot of mRNA expression. Each vertical line is a DNA point. This is a microarray that covers the entire genome. Each vertical thing is a DNA point. Consider it a proxy for a gene. Each horizontal line is an individual subject. Mother-reared, 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 peer-reared, 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 peer-reared. If it's red, it's overexpressed. If it's green, it's underexpressed. And what you see is virtually no overlap between mother-reared and peer-reared on these DNA points. If it's overexpressed in mother-reared, it's underexpressed in peer-reared. If it's underexpressed in mother-reared, it's overexpressed in peer-reared. With one exception, those of you with sharp eyes may notice that MR55, a mother-reared monkey, actually looks more like a peer-reared monkey. Turns out that MR55's mother was a neglectful, abusive mother. And we now know from studies at the Yerkes Primate Center, who study naturally occurring cases of neglect and abuse by rhesus monkey mothers, that their offspring look very much like peer-reared monkeys in that they have higher levels of cortisol, lower levels of 5-HIAA, higher levels of aggression, and higher levels of fear. Ron Barr mentioned some of our gene by environment work. Over the years, we have demonstrated that when you have polymorphisms in particular genes, especially candidate genes that affect emotional regulation, we get consistently gene by environment interactions. These are single candidate gene studies where we're comparing mother-reared monkeys and peer-reared monkeys who have one or the other of the alleles. So, for example, here's data from for the serotonin regarding the serotonin transporter gene. This is the serotonin metabolite, and what you get is a big deficit in 5-HIAA among peer-reared monkeys with the short allele, But the same short allele paired with mother-reared monkey maternal rearing gives you no difference from normal, perfectly normal maternal buffering. You see the same pattern with respect to aggression. Pure-reared monkeys with a short allele have high levels of aggression. Same short allele with a good mother, very low levels, as low or lower than what you see in their mother-reared counterparts. It's even more dramatic when you look at alcohol consumption. Here, if you're pure-reared and you have the short allele, you have higher, you drink to excess. But if you have that same short allele and you had a good mother, you actually drink less than normal. What may be a genetic risk factor for individuals with adverse early rearing may actually be a genetic protective factor for individuals with good early experience. And we see this pattern over and over again, not just for other dependent measures with serotonin transporter, but the same pattern we can see for other candidate genes, MAOA, dopamine transporter, DRD2, DRD4, mu opioid gene, neuropeptide Y, BDNF, COMT. In every case, what we see is when the less efficient allele is paired with adverse early rearing, we get deficits. When that same less efficient allele is paired with good early experience, you get either normal functioning or better than normal functioning. So here, for example, looking at a SNP in the CRH receptor gene, looking here at alcohol consumption, here's the less efficient allele. When paired with pure rearing, you get, again, excessive alcohol consumption, but that same allele, in combination with good early rearing, you get less than normal alcohol consumption. So what does this tell us? This tells us, among other things, that good mothering is pretty damn important and can go a long way to protecting individuals who carry genes, alleles that might otherwise put them at risk for adverse outcomes. In the presence of good early parenting, these individuals turn out to be normal or better than normal with respect to that function. So we've gotten very interested recently in what constitutes good mothering, especially in those initial days and weeks of life. And so I'm going to show you a couple videos to illustrate what I'm talking about. This first video is of a 2-month-old infant with its mother. The mother is low ranking. She has no clout in the social hierarchy. As the video opens up, there's a fight brewing to the right of the camera. The infant is interested in checking out the fight. The mother does not want the infant to go over there because if it gets in trouble, she doesn't have the clout to rescue it. So look what she does. Infant's looking over. Mother grabs the infant, grabs a flower, distracts the infant. There are the monkeys in the fight running away. Look at that face-to-face contact. Monkeys are not supposed to do that. Okay, the fight's over, coast is clear. Off goes the infant, and there goes the mother to follow up. That's an example of a really good attachment relationship. Here's another mother-infant pair. Now the infant is 19 days of age. And now they're actually doing face-to-face interaction. The reason this is astonishing, was astonishing to us, is that monkeys aren't supposed to do this. 50 years of studies of mother-infant interactions in rhesus monkeys, and nobody's ever reported this before. And the theories all say this is an exclusively human characteristic. But there you're seeing it yourself. So now we've studied this in more detail now that we know it's there, and it actually occurs fairly often. Here is mutual, in this plot, you're looking at the first month of life. Red line is infants' face-to-face interactions with other in the group, it's 0. But blue line is face-to-face interactions with mother. And here's mutual gaze, here's mother lip-smacking to the infant, here's infant lip-smacking to the mother, here's back and forth. And it's about 2 times every 5 minutes. That's a substantial amount of this for a behavior that's not supposed to occur. But look what happens. At the end of that month, it disappears. It starts dropping out, and it's gone by 5 weeks of age. Completely gone, out of the repertory. So what's happening at 5 weeks of age? That's the age that infants begin to leave their mother for the first time and start exploring their environment. And it's almost as if they don't need this anymore. They already have an attachment relationship with their mother. It's probably more important for them to be looking and paying attention to other social stimuli in their environment. And besides, they've got to get it out of their repertory, because if you want to start a fight when you're older, the best way to start a fight is to stare somebody in the face. So they have to get it out of their repertory, or it's going to get them in trouble. But there's something else worth noting. It's there at birth. It's there at day 1. So if you'll bear with me for a moment. Here's a 3-day-old infant, and that's not its mother on the right. That's Pierre Ferrari from Giacomo Rizzolatti's group in Parma, Italy. Rizzolatti is the guy who discovered or first characterized mirror neurons. And they wanted to know, Rizzolatti wanted to know how early you might get evidence of some mirroring activity. He didn't have any infants, we did. So Pierre has 30 seconds worth of still face, 3-day-old infant, first time they've ever seen each other. Now he starts opening his mouth, and watch the infant. The infant wasn't opening his mouth before. That sure looks like neonatal imitation, first described in humans by Andrew Metzoff and Tiffany Field 30 years ago, where they reported that newborn infants could imitate facial expressions of their mother or a caregiver within hours of birth. Here's another 3-day-old infant. This time Ken Vaughn is going to stick his tongue out. See it? See it? See it? 3 days old. Neonatal imitation. Let me play it again. So the infant starts imitating after the 4th stimulus. See it? So we've been making faces at baby monkeys for the last couple of years, and we now know a lot more about the phenomenon. First of all, just like in human infants, this reflexive aspect of it disappears very quickly. In humans, it's gone after a month or so. In rhesus monkeys, it's gone after a week. Given the fact that rhesus monkeys grow up 4 times faster than humans, it's right on schedule. Secondly, as in the human studies, not all infants do it. In the human studies, those infants that failed to imitate, the investigators threw them out of the study because they were screwing up the data. We're actually pretty interested in the infants that don't do it. In this case, these are nursery-reared infants on lip smacking on day 3. These are imitators. These are non-imitators. And in fact, over a fairly big population now, we find about 55% of nursery-reared infants can imitate during their first week of life. 45 cannot. Interestingly, when you look at mother-reared infants, and we get one chance to test them on day 3, when their mothers are put under mild anesthesia for their routine veterinary postpartum exam, we can leave the infants on their mothers, but we can still make faces at them. And when we do that, 95% of the mother-reared infants imitate. So already, by 3 days of age, there's a major rearing condition difference. So does it make a difference whether you imitate during that first week of life or not? Well, we followed some of these infants up. We now know that over the first month of life, they have much better sensory motor coordination as indexed by a reach and grasp test where you put an object out, infant has to go and reach and grasp for it. Imitators do significantly better than non-imitators on that from the very beginning, and that difference is maintained. When they get a little bit older and they get to the age when they should be starting to play, imitators, those who imitated during that first week of life, now play twice as much as those that did not, particularly during months 4, 5, and 6, when play is really starting to take off in these monkeys' repertory. Perhaps most intriguingly, when they're a little bit older, 2 years of age, and now have been living in stable social groups for a long time, non-imitators show much higher levels of self-directed behavior, clasping their bodies, sucking their thumbs, than the monkeys who could imitate during that first week. And perhaps most intriguingly, those monkeys who failed to imitate during that first week of life, by 2 years of age, a fair proportion of them have developed autistic-like stereotypic behavior patterns spontaneously. None of the infants who imitated during that first week show any of these autistic-like stereotypies. We just might have stumbled on a very early screen for risk for developing autistic-like stereotypic behavior patterns, at least in monkeys. So what does this all mean, and where is this going? Well, for sure, we're going to be following up these infants as the successive waves of newborns come into the lab. We're going to follow them up, see what happens as they see if these early patterns of less play and more self-directed behaviors are repeated in subsequent generations. Our first batch of infants are now old enough to have kids. The mothers are old enough to have kids of their own. We want to see if this capability is transmitted to the next generation. We want to take those infants who are not imitating at day 3 of age and see if we can get them to start imitating with interventions, giving them more social stimuli, the kind of stimuli that they're missing by not being with their mothers. We have just started eye tracking data. Had it been a week later, I could have shown you a slide that at least now looks like imitators focus, Chuck, on the eyes and the mouth during imitation patterns, but the non-imitators only look at the mouth. And finally, we want to look at brain function. The initial studies of mirror neurons by Rizzolatti were done with single-unit recordings and adult macaques strapped into chairs. There's no way I'm going to do anything like that with my baby monkeys. But it turns out that you can use EEG. And down the road from NIH is the University of Maryland, where one of the world's experts in human infant EEG is, Nathan Fox. We now are collaborating with him. We have these snazzy little caps, just like they have in human infants, with electrodes snapped on. And our first batch of data suggests that there is a distinctive EEG signature during periods of imitation. What it is desynchronization of mu rhythm at low amplitudes. So low amplitude mu rhythm desynchronization. When you give these monkeys a rotating disc to look at, non-social, you don't get desynchronization. But when they're looking at either a lip smacking or a tongue protruding model, you get that desynchronization. And when they're performing those same actions, you get that same pattern of desynchronization. This is the anterior electrodes. These are posterior the parietal electrodes. You do not see this pattern when the monkeys are not imitating. You do not see this pattern in monkeys who fail to imitate. So one of the things we want to do, if we can restore the behavior with intervention, do we also restore this pattern of EEG? At any rate, it's quite clear that this work is going to go on, and we think it's leading into some interesting directions that shows to us how really important those first few days of life are. We used to blow them off, not anymore. These are some of the people who contributed to the imitation project. These are some of the people who contributed to the larger research body. I thank all of them for their effort, and I thank you for your time.